Physical maps. As in previous topic, we discussed that in case of genetic linkage mapping, the exact location of the genes, it cannot be pointed out. But on the other hand, this is not the situation for physical maps, because it can pinpoint the physical location of the genes and as well as the genetic basis distances between the different landmark. So on the basis of the number of nucleotides, it can pinpoint that how one genetic marker it is separated from another. तो फिजिकल मैपिंग में ना सिर्फ हम जीन्स की लोकेशन को फाइंड आउट कर सकते हैं, बट इन टर्म्स ऑफ नंबर ऑफ न्यूक्लियोटाइड बेसिस हम ये कह सकते हैं कि एक लैंडमार्क जो है या एक जेनेटिक मार्कर जो है वो दूसरे जेनेटिक मार्कर से इतने बेस पेयर सेपरेटेड है. सो व्हेन अगेन वी विल कंपेयर these two type of maps. So in case of genetic linkage mapping, the exact position or actual position, it cannot be elucidated. The reason is that the pattern of inheritance, that due to variation in the recombination frequency, the exact position cannot be pinpointed. कि जो रिकॉम्बिनेशन की फ्रीक्वेंसी है वो एक जो जीनोम का पार्ट है वो डिफरेंट तरीके से रिकम्बाइन करता है एज कम्पेयर टू अदर पार्ट. सो बिकॉज ऑफ दिस रीजन द एग्जैक्ट पोजिशन कैन नॉट बी पॉइंटेड आउट. Another reason was that in case of genetic linkage maps, basically the traits were studied and genes were not considered. Now in case of modern maps like the physical maps, now the genes, now the traits, they are characterized in terms of genes that help to designate the position of particular genes. Now different type of mapping techniques are available. And here in case of physical maps, any clone within the genomic library, it can act as a starting point. For example, if we want to map a certain genomic DNAs, then it can be digested, then it can be recombined in the vector molecule like plasmid, bacteriophages, M13 vector, cosmids, plasmids. So they can act as vector. So we can develop the genomic library. We can pick any clone, we can sequence it, and then we can find the position of other genes. And if we have some longer length of the target segments or the restriction fragment, then it is more helpful. Like now we have bacterial artificial chromosome that can minimize the number of fragments that are generated due to partial digestion, so that more than 100 kilobase fragments, they can also be cloned. Although some other vectors like the yeast artificial chromosomes are also available that can clone larger DNA fragments, but they can introduce certain addition, deletions during the process of replication. So they may not be so much suitable, but overall we can say that certain clones, they can act as a starting point to construct physical maps. And later on we will also study that some other characteristic feature like the polymorphic sequences within the genome, and that form the basis of the study of restriction fragment length polymorphism. It can also be used to construct physical maps. So a variety of methods can be used to study the physical maps, and you should be aware that in coming lectures mainly we are going to focus on the physical maps. And before going to onward, one should know the difference that what are the genetic linkage map and what are the physical maps.